Dear students, today let us learn about micropropagation. Let us start with the introduction. Micropropagation or in vitro clonal propagation is a rapid method for vegetative or asexual multiplication of genetically identical copies of a cultivar using tissue culture technique. Using this technique, in vitro clonal propagation can be achieved in a short time in a small space, and large numbers of plants can be produced starting from a single individual. The use of tissue culture technique for micropropagation was initiated by G. Morrell in 1960 for propagating orchids. Let us now see the general procedure of micropropagation. The general process of micropropagation is a complicated process and requires many steps or stages. Murasige in 1978 proposed 4 distinct stages, that is, stages 1 to 4 of micropropagation or in vitro clonal propagation of plants. Out of these 4 stages, stages 1 to 3 are usually carried out under in vitro conditions, whereas stage 4 is accomplished in greenhouse condition. Further, Dewerg and Maney in 1981 suggested an additional stage 0 for the process of micropropagation. Let us see the stages of micropropagation. First, stage 0. This is the initial step of micropropagation in which stock plants used for culture initiation are preconditioned before culturing in vitro. It is usually done by growing the stock plants under carefully monitored conditions at a relatively low humidity for at least 3 months, and also includes measures adopted for reduction of surface and systemic microbial contaminants of the explant. Next, stage 1 or explant culture initiation. Murasige defined this stage as the first in vitro stage for micropropagation, and the main steps involved in this stage are preparation of the explants, including surface sterilization and initiation of culture. Surface sterilization of the explants includes procedures to reduce surface microbial contaminants and also to control microbial contamination within explant tissues, which could not be eliminated at stage 0. For micropropagation, explants with preformed vegetative bud such as shoot tips and axillary buds, which are suitable for enhanced auxiliary branching, are usually selected for culture initiation. Initiation of culture involves culturing the explants on suitable culture media, followed by subculturing a few times to see whether the microbial contaminants are fully removed or not. Next is stage 2. This is the stage for multiplication of microshoots. This stage is the major stage of micropropagation process and involves multiplication of shoot buds using defined culture medium that stimulates maximum proliferation of microshoots. The main approaches followed for multiplication of shoots in this step are: first, micropropagation by axillary and apical buds. This approach involves multiplication through the growth and proliferation of apical and axillary shoot meristems present in the explant. When the shoot tips containing axillary shoot meristems are cultured on media containing cytokinins such as 6-benzyl aminopurine or BAP, kinetin and gelatin, the axillary shoots of the explants develop precociously, which proliferate to form clusters of secondary and tertiary shoots, which can be further subdivided into smaller clumps of shoots and multiplied when subcultured on fresh medium, ultimately leading to extremely impressive rapid clonal propagation level in the range of millions within 1 year. The next approach is micropropagation by adventitious shoots. Shoot multiplication by this method involves the induction and multiplication of adventitious meristems through the process of organogenesis or somatic embryogenesis. The meristematic regions present in the explants such as stem, bulbs, combs, tubers and rhizomes etc. regenerate multiple shoots directly on explants other than at normal leaf axial regions when cultured on a suitable culture medium. Continuous propagation by adventitious shoot proliferation can be achieved by splitting the shoot bases and subculturing them on fresh medium. Clonal propagation by adventive embryo formation is another useful approach for many important plant species where adventive embryos can directly arise from a group of cells within the explants or from primary embryos like at the tip of leaves in vitro in orchids or from the nucellar tissues of citrus and mangifera. The third approach is micropropagation by indirect organogenesis. It involves multiplication of chelae derived from organs, tissues, cell or protoplasm and induction of shoots from this chelae by either organogenesis or somatic embryogenesis. These birds can further be multiplied following the procedures A and B described above. It can be the fastest method of shoot multiplication and cloning of plant species. However, cultures in which chelae are produced tend to be of low value as the means of micropropagation, since such clones commonly exhibit semiclonal variations due to the genetic instability of their cells. Now the fourth stage is stage 3 or the rooting of microshoots. The in vitro regenerated and multiplied shoots produced in stage 2 are transferred to a rooting medium in this step to obtain complete plantlets. The media used for rooting normally contains less salt concentration and includes growth regulators like auxins, which promote rhizogenesis in the microshoots. Sometimes, shoots of certain species may be directly established in the soil and root them in vivo or ex vitro like microcuttings. In such cases, stage 3 is usually skipped. The last stage is stage 4, which is acclimatization or hardening of. It is the last step of micropropagation and comprises steps taken to ensure successful transfer of plantlets of stage 3 from aseptic culture environment of the laboratory to the environment of greenhouse, and the process is known as acclimatization. Since the plantlets growing in the culture vessels were growing under 100% humidity inside the vessels, and the plantlets usually have no wax and poor stomatal response, they need to be acclimatized to ensure successful survival outside the vessels. Therefore, acclimatization is carried out by supplying bottom head aids to pots with plantlets or cuttings and maintenance of a dense fine particle fork system within the greenhouse to enhance the rooting process. The plantlets can also be established in the artificial growing media such as soil-less mixes, rockwood plugs, or even sponges. Next, let us take up the factors affecting shoot multiplication and micropropagation. Morphogenesis and proliferation of shoot buds depend on various factors influencing the relative incidence of organogenesis or embryogenesis. Like, the first factor is physiological status of the plant material. Generally, explants isolated from more recently produced parts of the plant are more regenerative than those from older senescing regions. The second factor is culture media. Shoot multiplication medium for micropropagation generally contain auxin and cytokinins, and organ differentiation is regulated by the ratio of these two growth regulators. However, cytokinin is usually omitted and auxin is added in the rooting medium to induce rhizogenesis. The presence of gibberellic acid or Ga and abscisic acid or Aba in the medium inhibit root formation, while activated charcoal may induce the formation of adventitious roots in some species. The third factor is the culture environment. The intensity and quality of light also controls the organogenic differentiation and growth of shoot in cultures. In most of the plant species, the optimum light for shoot multiplication is 1000 lux, and blue light at 467 nanometer induces bud formation, while red and far red light induces root formation. A diurnal illumination of 16-hour day and 8-hour night is generally found satisfactory for multiplication and proliferation of shoot, and most micropropagation cultures are normally maintained at 25 degrees Celsius. The fourth factor is genotype. The regeneration capabilities of explants and culture differ depending upon the genotype of the plant. Therefore, the micropropagation system developed for one particular cultivar will not automatically be applicable to another plant, even within the same species. The next factor is in vitro rooting. Media having low concentration of salt and a suitable auxin are satisfactory for rooting of micropropagated shoots. And the last factor is acclimatization of plants transferred to soil. Micropropagation on a large scale can be successful only when transplanted plants show high survival rates and costs involved in the process is low. Next, let us take up the advantages and limitations of micropropagation. First, let us see the advantages of micropropagation. There are many advantages of micropropagation or in vitro technique of plant propagation compared to the conventional procedures, which made this technique to become a routine procedure for large-scale production of many plant species including crop plants, vegetables, spices, medicinal and aromatic plants, etc. The significant advantages of micropropagation are: first, it requires relatively small growing space. Next, through tissue culture, over 1 million plants can be grown from a small, even microscopic piece of plant tissue within 12 months. Next, shoot multiplication usually has a short cycle, that is 2 to 6 weeks in micropropagation, and each cycle results in logarithmic increase in number of shoots. The fourth advantage is, since micropropagated plants are genetically homogeneous, plants which are more or less true to type to the parent plant can be produced by this method. Next, since shoot tip culture allows the plants to become virus-free, disease-free plants can be produced from diseased plant stock by micropropagation. Tissue culture techniques gives propagules such as mini tubers or microcombs for plant multiplication throughout the year, irrespective of the season. Next, the small size of propagules and their ability to proliferate in a soil-free environment facilitates their storage on a large scale and allow their large-scale dissemination by suitable means of transport across international boundaries. The next advantage is stocks of germplasm can be maintained for many years in vitro. Next, clonal propagation in dioecious is extremely important since seed progeny yields 50% females and 50% male plants, and only one sex are desired commercially. Propagation of these plants can be achieved by micropropagation. Let us now see the limitations of micropropagation. First, requirement of sophisticated facilities for micropropagation is the first limitation of micropropagation. Next, micropropagation demands greater skill in handling and maintenance than conventional techniques. The third limitation is shoot-tip derived plants may show genetic instability. Example, 6 to 35% of banana clones developed through shoot-tip culture show morphological variation. The next limitation is poor adaptability of most of the in vitro raised plants to soil or acclimatization is the major factor limiting the widespread use of micropropagation technique at the industrial level. Let us now see the conclusion. Micropropagation is a rapid method for clonal multiplication of genetically identical copies of a cultivar using tissue culture technique, and in vitro clonal propagation can be achieved in a short time in a small space, and large numbers of plants can be produced starting from a single individual compared to the conventional procedures using this technique. The many advantages of micropropagation or in vitro technique of plant propagation has made this technique to become a routine procedure for large-scale production of many plant species. Thank you.